remove the rest one, or keep top 10, remove the rest. So now your requirement becomes small, your field requirement is small, your labor requirement is small, your time requirement is small, everything becomes small. But how you are going to be able to tell which plant is best yielded, or which plant is resistant to some disease, or which plant is resistant to fire? Whatever character you want. We want to be able to tell at the seedling stage only. That is possible if we have some kind of markers. We have some kind of markers. Okay. Like in our case, our names are our markers. Okay. So like that, if you have some kind of marker, so conventionally about 30-40 years back when I was a student, that time DNA was still something we imagine only. It was not possible to visualize DNA. So that time the markers were morphological markers, like color of the seed, presence or absence of home. These kind of things were morphological markers. And if some of these were linked with some of the traits of agronomic interest, the period was very large. Okay. Then came protein markers, like isotypes or storage proteins like that. Then DNA became extraction of DNA, and manipulation of DNA became very common. And the marker came, there was RFP, restriction fragment-like polymorphism. Then PCR came, and as PCR came, the marker was rapid, RAPD, on AFL. Okay. Then when sequencing became possible, SSR marker. And now ruling the roost is SNP markers, SNP single nucleotide marker. So now if you can link these markers to some trait of your agronomic importance, you can look for that marker when in the seedling stage itself, and you can then you don't need to grow the crop through the whole season or whole. So what basically happened? Still you are selecting, but now you are selecting in a smarter way, right? Still you are doing what you are selecting. Only your selection is getting smart. So this is the QTL is not a gene actually. QTL is quantitative trait locus. It's a portion of DNA on the chromosome which is linked to a particular quantitative trait in plant height. These are quantitative traits. These are not governed by one gene. So they behave like gene. They can be identified like gene, but they are not genes. So to be able to do this, we have to be able to link some QTLs with the trait. And to do this, the classical method is biparental mating. This is a work in our lab. One of our students, because all of my students have worked in this, but mainly this is work of Rahul Kalrate, in which he took two varieties, Disang, which is a yielding variety, and Konyahu, which is a drought-tolerant lentils. Crossed these two, Konyahu and Disang, he crossed these two, got F1. He verified that F1 using some markers that these are F1 because he used many markers which were identified Konyahu as well as Disang. So when he got both the markers, he could show that these are F1s. And then he raised F2, F3. In F3, he did SNP genotyping, assembly genotyping or SNP genotyping using GBS technology. There are various technologies. One is through hybridization with chip. Another is GBS. GBS means genotyping by sequencing. Because now genotyping has become very easy and very cheap. we can simply genotype the population, sequence the population. Sequencing has become very inexpensive. You can sequence the whole population at a lesser coverage, of course, and then from there you can identify the SNPs. These are statistical methods and very compute intensive methods. You need good quality compute and good knowledge of bioinformatics. You will remember in my inaugural lecture also I was talking about bioinformatics. This is because if you want to do this kind of work, bioinformatics is required. And then these populations were also phenotyped. Phenotype means they have phenotypic traits were studied. Okay, and then by combining the genotype and the phenotype, we could identify various QTLs. These are the markers we could identify on each chromosome, each line. These are all chromosomes. This is chromosome 1 to like this up to 12. Rice has 12 chromosomes. Twice n is equal to 24. And each line is a marker, SNP marker that we identify. And these are some of the QTLs. QPH1 means this is related to plant height. This QTL is for plant height. One means chromosome number 1, and this is the position 29. This is the left marker, this is the right marker, and this is the percentage of phenotypic variance that QTL is doing. Like this is the list of QTLs. So QTLs can be identified linked to a particular trait through biparental mating. Biparental mating means you have to have two parents of distinctly opposite character. Cross them, take the generation forward, genotype it. You can genotype in many different ways. Before him, the other Rahul, senior Rahul, who is our project scientist, who has been, I think, working with you all the time, he did similar work with SSR genotyping. Okay, genotyping can be done in many ways, but nowadays SNP is the easiest and the most informative and most fastest. Okay. So if you can link this kind of identify this kind of QTL, then the QTL is tagged by some markers, and by looking for those markers, presence or absence of those markers, you can determine whether your variety has the particular character that you are interested in or not. The plant in the population, and if it is that, you keep that. If you don't have, you just throw that. So your selection has now become smarter. So once you find the QTL, since QTL is not a gene, inside the QTL there may be few genes or there will be many genes depending on the marker distance. If the marker distance is very high, there will be many genes. If the marker distance is low, there will be few genes, but there will be a lot of genes. Now, because rice genome is already known, rice genome has already been studied, sequenced, resequenced again and again, and lot of functional genomics work has also done with rice. Okay, there are some websites. I did not put the slide. I should have. I can give you afterwards a handout or something. There are some websites where you can get all the information. Like Gemini is one. Gemini is one website where you get all this information. Then the Japanese rice portal, that is one. Like there are some websites. So since you know the whole rice genome and you know which genes are present in where, and you have now the two flanking markers for the QTL, you can identify what are the genes inside the QTL. Okay, and by doing a literature, simple literature study, you can make an educated guess that which of these genes, suppose there are 100 genes inside a QTL. Now out of these 100 of genes, which gene may be giving me the character? Because simple literature study. Suppose it is doctorants, and I find a gene in my QTL which is related to water movement. Okay, so now I can make an ethical guess that okay, this gene may be responsible for giving me drought tolerance. Okay, another gene is suppose related to high metal tolerance. That can also give me drought tolerance also. I can make some idea. And then I can try to prove or disprove that these are not candidate genes, these candidate genes are actually giving me the trait or not. And this has become very, very important, identification of candidate genes and editing candidate genes, because with now genome editing becoming legal, it is not necessary to go through all the marker-edited selection thing. If you know the exact gene, which gene is giving me this trait, which allele is giving me this trait, because this all is rice for all of the same crop. One is tolerant, one is susceptible. Both are having the gene, only the allele is different. Allele is different means some nucleotides are different, or some nucleotides may be missing, some nucleotides may be more. Only. So by genome editing, if you can change that, you don't need to go for all this marker-assisted breeding and all that. Okay, this was very simple. And 3 years back, one lady named Sarah, here she gave a lecture about developing bacterial leaf blight resistant rice through genome. 3 years back, in this platform. That time it was not free of regulatory process. Today it is free, so maybe that line will come into mainstream very soon. But for this, the method of QTL identification that we just now discussed, you need to have a recombinant inbred line population. That means when you make a biotechnological thing and you enhance this, you call it recombinant genes, recombinant inbred lines population. But suppose you have a population of samples, like in Titapur, yesterday you went to Titapur, and somebody must have told you that they had about 4,000 accessions of samples collected from all over Northeast. Right? Now you have samples like that, and you want to find a gene or a QTL from there. How many biparental cross you make? If 4,000 lines you want to cross with each other, this will be billions of combinations. Okay, will not possible. So next smarter way came is marker trait association or genome-wide association studies. Okay, GWAS or marker trait association, in which basically linkage distortion is taken as the fundamental principle. Now, all of you have idea what is linkage? In a chromosome, when two genes are close together, they tend to move together and you call them link. Right? So the same principle, you can say the QTL in this case is linked with the trait. Linkage. They tend to move together. Now, normally if a population suppose I have 1,000 plants, 1,000 different lines in a population, I am growing them. And if a trait and the QTLs are not linked, they will be evenly distributed. Evenly distributed. Right? But if these two are linked, then their distribution will be, they will move together. Whenever you find a plant with that particular marker, you will also see that trait. Right? So your linkage equilibrium, when this is distributed equally, it is called linkage equilibrium. The linkage is in equilibrium. Everything is equal. But when there is a linkage with the trait, then the linkage equilibrium has got distorted because these two are moving together. They are not equally distributed anymore. This is called linkage equilibrium distortion. Okay? And this is a concept that I find very difficult to teach my PhD students also. So don't try to think that you can understand it very well, but if you can get the general idea of the principle, I will be very happy. Okay. So your linkage has got distorted if it is linked. And if you can identify that linkage distortion, you can identify the QTL which is linked to your trait. Okay. Use statistics. You have to be able to genotype the whole population. so that you can identify the markers where they are present. You have to be able to phenotype the whole population, okay, so that you get the phenotypic trait of each of the plant, and then you have to match this phenotype with the genotype using statistical method, which will tell you where is the linkage distribution, and the linkage distribution distortion you can see from this graph. This is called a Manhattan plot. When you do genome-wide SNP study, the computer gives you a Manhattan plot. Without computer, you cannot think about doing this manually. But once you learn the principle, the people who are working with GWAS, some of my PhD scholars are here, I will not name them. How many of them understand this principle? I have serious doubt. They should do serious effort, take serious effort to understand this principle. This is a baseline. Below this each dot is a linkage pattern. Linkage association with the linkage. As long as it is below the baseline, this is in equilibrium. When it is above the baseline, like this one, like here this one, like here this one. When this goes above the baseline, that means this is linkage disequilibrium. And now you identify that one, that is a marketic association. Okay, and then you look for candidate gene there. So using genome-wide association studies, we have identified various marketed association for drought, under drought condition, under control condition. You have to go under both conditions. Suppose you want to find out a gene, or suppose in this case it is drought tolerance, or suppose BLT, resistance to BLT, or resistance to blast. So you have to grow the crop under BLT infestation condition and under control condition, so that you can make a contrast. So, and you will get QTL both from the stress condition as well as the control condition. Some of the QTLs were identified. How much time I have, Sunita? 10-15 minutes? Yeah. So, our study gave us QTLs like this. And when QTLs are bunched like this in one place, this is more likely to be a real QTL. Because any QTL, because the QTL has several genes, it will be linked to several traits of similar traits. So this is not a gene, this is a QTL, which has several hundreds of genes there. Okay, so suppose you are looking for drought. So what can be important to drought? Suppose water absorption can be important to drought. Good light can be important to drought. Stomata opening closing can be important to drought. Okay, so if a QTL is linked to stomata opening, the same QTL has some linkage towards suppose root line, that means this is a very good QTL for drought. Okay. So I think yesterday Titabar, this what might have been explained, how this Titabar and we together we developed Ranjit someone. I think this work was explained and I will not go into any detail. This one. This is the same Ranjit someone development work. Now next comes genome assisted breeding, genomics assisted breeding, or genomic assisted selection. Marker-tagged association breeding, marker assisted breeding, marker assisted backcross breeding, or all these are also genomic assisted breeding. But if you see the genomic assisted breeding in a whole way, this can be summarized in this model. You have to have genetic resources, which can be biparental mating, which can be a multi-parental population, just like I talked about in GWAS, or you can have a natural population. Okay, these are the residues, and this has to go through high throughput genotyping, or for that you need a sequencer. Nowadays, Illumina is the sequencer of choice for this. I have shown a sequence, Illumina sequencing machine here. where you will be sequencing, resequencing, you can do microarray, chip-based microarray. All these are techniques by which you can do SNP identification. Okay, you can do SNP identification by sequencing, resequencing, GPS, or you can use chip-based microarray and genotype. In chip-based microarray, already the SNPs will be there. It will be a small chip where millions of SNPs will be there. You will hybridize that with your DNA and find out which of them are hybridizing to know the SNPs present in your line. Then you have to phenotype. Nowadays, phenotyping also has become very high throughput. Normally, you do phenotype by going into field designing everything. But nowadays, you can use drones, unattended aerial vehicles. You can use phytodrones, like we have one. I think somebody has taken you around the phytodrone facility. You can do phenotyping inside that. You can have rain-out shelters, so those are basically. Or you can have this kind of vehicle which moves on top of the crop canopy and it has all kind of imaging devices attached. We can take those pictures which can be studied by computer. There are many ways by which you can do phenotyping. What you have to do for phenotyping for yield, quality, disease resistance, biotic stress, plant architecture, all these traits you can do phenotyping. And then you do marker-trait association, either by linkage mapping or GWAS or genomic selection or marker-trait selection. You do this and ultimately you get improved variety. So what is genomic selection? Now the selection has got still much more advanced. Where genomics come into play? You have a training data set. Means what? You have a small population which is representative of the bigger population. Like suppose in this class, this is a population. I take 10 students from here without any bias. I should not be biased that I will take only girls or I will take only boys. I should not be biased that I will take only the tall people or short people. No bias. Randomly I take 10 out of you. Now this is a representative of the whole population. What I do, I study all the phenotype in the 10 population. So phenotyping is the costly part. Genotyping is cheapest. Phenotyping is the most expensive. So instead of phenotyping the whole population, I phenotype only this 10. And then I do the GWAS, the marker-trait association in this 10. I know that this marker relates to contact, this marker relates to gender, this marker relates to intelligence, like that. And now I genotype the rest of the population. I genotype the rest of the population. Don't phenotype. By just this association that I have found, I now predict. Oh, he has this marker, so he must be Tom. Or he has this marker, he is not a he is a she. Understood? Prediction based on genomic data. So now this is the stage where this is happening. Okay, you don't need to phenotype the whole population. You phenotype only a representative sample of the population, but your representative sample should be representative. It should not be biased. There should not be any population structure to this. And you genotype very well. And you have to have good bioinformatics skill so that you can identify the associations. Okay. Now you can predict. You can do genomic prediction. You can predict. And based on that prediction, you select the best among that. Even it is, I've just given an example in plants. You have a training population, and you have a target population, which is very large. Just because of space, I have shown it to this is very large. I do all kind of study here, and then put the study. I do develop a model here, something like this. Y is equal to, suppose Y is equal to yield. Now X into B plus Z into N plus C, something like that. This is a simple regression model. Any one of you know statistics, you will know this is a simple regression model. You know what is regression? How many of you know regression? Correlation, regression. Yeah. One hand in the back. Correlation is when two traits are dependent on each other. It may be positively correlated, it may be negatively correlated. If when a character moves, the other character also moves. If same direction, it is positive correlation. If opposite direction, it is negative correlation. It's correlation. When one character is dependent on other character, the other character may or may not depend on this character. Suppose if is dependent on number of seeds per plant, 100 seed weight. Okay, but 100 seed weight is not dependent upon yield. If yield increases, 100 seed weight may increase, may not increase. But if 100 seed weight increase, yield will increase. Right? So now this is regression. Yield is dependent upon 100 seed weight, number of seed per plant, number of pillar per plant, length of panicle, many characters. Okay. Now you can develop a model. If you can do this type of study, you can develop a model. So this time seed, number of seed per week plus this time of X time of number of pillars, Y Z time of this particular character will give me this amount of it. This is called a regression model. So basically what you will be doing is you will develop a regression model on your interest population. Okay, and now by fitting the regression model on the genotypic data that you have from your target population, you will just predict what will be their yield, what will be their reaction to a particular disease or particular stress. Okay, this is where plant breeding is at now. So starting from undomesticated, when the lady had to sow some seed because she did not have any food, millions of years ago, we have arrived at the stage whereby genotyping and phenotyping a small population, small intelligent population, we can predict the behavior of the whole population and select the best genotype. Thank you. Any questions? No question means either you have understood everything, which is almost impossible, or you have not understood anything. So which is most probably the latter of the case. And if that is the case, I am a bit poor teacher. Yes.
So how we confirm that it is exactly a single nucleotide mutation or just because of the how we confirm that this is
that comes from the coverage. You never sequence only a single copy. You sequence many copies and you stack them together. Suppose you have sequenced 10 copies. And in 9 cases, suppose this shows a mutation, one doesn't show a mutation, you know that it is a mutation. The coverage gives you the confidence. Okay. Depth of coverage gives you the confidence. Any more question? Yes.
So what will happen when we select a population from that main population advertised?
If it is biased, then your regression will never fit you. Suppose I have selected only the ladies. Now how that regression will fit with the boys? It will not fit. It will never fit.
Biparent population, markers are the same for the one variety as well and for the second.
So, in biparent meaning the signature is different. I am talking about biasness in the selection. In case of GWAS, especially in case of genomic selection. In case of GWAS also, there should not be a population structure. The population should be evenly distributed. The Poisson distribution should be there. Okay. If there is a structure, then you have to take statistical measure to there is no blackboard here, otherwise I would have shown you the diagram. You have to take measure, statistical measure, that you can remove the biasness as much as possible, which is not completely possible, but as much as possible. China? Yes, Sona.
Is the taking a representative group same as making a co-collection?
Yes, of course. Yes. See, what he is asking, when you have a, suppose, example like this, we have 4,000 examples. Now, genotyping 4,000 examples, phenotyping 4,000 examples becomes expensive. But if you can select about 400 from there, which has almost every character, the whole 4,000 population is. Now we call this is a code collection. Okay, and we can now extensively genotype and phenotype this code collection, and this data can imply to all number of the whole population. So this is called a code collection. This is simply like in case of genomic selection, we take a 10%. In case of genomic selection, we call this a 10%. Okay, this comes from, again, artificial intelligence, neural network. In artificial intelligence, artificial intelligence, have you heard about artificial intelligence? Any idea what it is? Intelligence cannot be artificial, no? Intelligence is real. It is in your brain. Why call it artificial? The computer is shown a pattern again and again and again. Shown means I don't say visually shown. It is exposed to a pattern again and again and again. So that the computer knows if this is the pattern, this is the outcome. Okay. And when you give a new data set, it looks for the pattern. And if it says pattern A, it says that this is the outcome B. If it says the pattern C, this is the outcome D. Like that is artificial intelligence. Basically, you have trained the computer to identify pattern and predict on that. Same thing what we are doing here. Here is the training data set. You have a training population. In the training population, you have done all the work, hard work. And that pattern you are imposing on your whole population. Yes.
We found some marker trait associations. Like for us, some of them may be novel and some of them may have been reported earlier. So how do we go for validation? Can you just explain?
This is not the time and place for you to answer that. Ask that question. Okay, you have to answer that question and tell me.
Yes. But you are working in that same lab.
Says my PhD student going to submit in 1 week. That is why I won't answer this question.
I have a question.
Yes, Malay.
So why was it called Ranjit support? Is there a specific system of nomenclature for every variety?
Now, Ranjit is a very popular variety in Assam. Most popular variety, mega variety, which covers about 1 million hectares, more than 1 million hectares in Assam. And also it has gone into other states, neighboring states like Bihar, etc. Now we have just put the sub-1 trait in the Ranjit. Nothing else. Ranjit has all its trait intact. Only it has got the additional sub-1 tolerance. Now if we change the name, people will see the new variety and adoption will take time. But keeping the name Ranjit, just adding sub-1, we have tried to, this is just a branding mechanism. Okay, just like you say, advertisement, you get a fair and lovely and then you will try to make this special. Fair and lovely X or something like that. Same thing with the added something to give it added value. We have just given a brand. So that is original brand name, but the added value is coming. There is a very official name is very different. And that is very complex. Like we have to write AAU, Dhan. So just to start with AAU, the university, then Dhan, signifying as a rice variety, then some numbers, where the cross number and all that is a complicated.
So there is this.
Yeah. This is like this nickname and proper. Okay. Nickname is different, by which everybody at home calls it. So the Ranjit symbol is the nickname. The proper name is different. And in Assamese Brahmin families, the actual proper name nobody knows. It is only in the what is called Pusti. Morning. So it is only there. So similarly, our name is only the whole. Property.
Sir,
Yes.
sir, actually, I have some doubt QTL. As you have said that QTL may be a collection of functions that move together to here. So, if for one specific condition, suppose top or right and right. So, only one QTL is responsible, or more than one QTL can be responsible.
You did not notice very well. I say QTLs we identified give about 10%, 12%, 15% phenotypic variance. That means what? That particular QTL was responsible for 12% of the trait. It was giving 12% PD, phenotypic variance. It was responsible for 12%. But trait is 100%. So there must be other QTLs. And these are quantitative traits. Quantitative traits are where many genes give small, yeah. The word is not coming to my mind now. They contribute in a small manner to give the ultimate trait. Plant height, yield, these are all quantitative traits. Thank you. Yes.
Sir, my question is related to hybridization method. There are a lot of the methods, predictive method, bulk method, multi-cross.
All those are selection methods. Don't confuse. Hybridization method is simple. You take two progeny, decide which one will be female, which one will be male. You remove the male organs from the female and put the pollens on the female stigma. That is hybridization.
Which one is better, or how can we decide we should go with which?
You have to do a whole primary course for that. Each method has its own advantage, its own disadvantage. Okay. Depending on your scenario, first thing you have to see whether your crop is self-pollinated crop or cross-pollinated crop. Then how much land you have, how much space you have, how much money you have, will determine which selection method you follow. Okay? Each one, this is a very complicated, very time consuming. But yeah, that is a very interesting and good question. If you want to be a good plant breeder, you must know the methods and you must be able to select the method.
Yeah, any questions from participants? I think we have to organize a few more lectures of yours because our students are mostly asking questions.
Yeah, that is right.
You have to take more classes. No questions? Thank you. Okay, she has one.
Because of the closeness of location, but what is the reason of linkage disequilibrium? Because the creature will be from different chromosomes. Then how it leads to linkage disequilibrium? That is the reason.
No. Linkage, don't mistake the linkage between genes here or linkage between QPLs. It is linkage between the QPL and the trait. Just like the trait, the chromosome, genes are linked. The trait is also linked to the chromosome, the trait. Trait is also linked to the QPL or the gene. Now, if that is linked, then your linkage will, this linkage will show a different picture than if it is evenly distributed. Suppose if it is evenly distributed, then maybe 2% or 1% of all the time will show where both this will come, the QTL as well as the trait. But if it is distorted, instead of 2%, in 10% of time or 15% of time, you will see this linkage. That means this linkage is distorted. This is not equal to the average linkage, like in this. Just take 1 minute. Okay?
Yes, yes, yes.
Like here, this is the average linkage. Up to this is the average linkage. Up to this means we are taking the outliers also, extremes also. Up to this is the average linkage. Above this is more than average. So this is now linkage equilibrium distortion. Okay, LD linkage distortion. Okay, and now this one you can take as a.
the doubt is like in linkage, it can explain like it's close together, means that alleles will be close together or
Because of the mass, I am not able to understand you at all.
in case of linkage, we can explain like it will be close together, means those locations, whether it is marker or trait, it will be close together, so there is less chance of recombination. Likewise, we can explain in case of linkage. Then what is the phenomenon behind the
See,
linkage?
a trait, suppose, sit down, suppose some other students, the gene sub C is giving the trait. Now, wherever someone C will go, that will be destined to someone else, to someone else. Right? That is the linkage. Because the gene gives that character, no? The gene produces that particular enzyme or that particular situation whereby that character develops. So that is the linkage. That is the linkage. So wherever that gene will be, that will be there. In case of substance tolerance is a G, is not a QTL. Though it is called QTL, it is a G. It gives 70% of the trait. Someone C gives 70% of the trait. Okay, so you see wherever someone C is there, it is someone's point. But for other, it is not like that. Because they give 10-12%, so that effect is sometimes masked also. So you will not see all the time wherever this QTL is there, that character. But you will see more than normal. Okay. You discussed this in your molecular building class, because she is a PhD student.
Yeah, she's a graduate. Thank you, sir. Thank you for the nice presentation. After student understood about what molecular markers is and how molecular markers can be used for crop breeding. Thank you once again, sir.
Thank you, Sumita, for giving me the opportunity. And thank you, students, for looking at present here. Thank you.
So you have a cup of tea and then want to see the practical session down. Good. Then we have a talk by Dr. Manoj Prasad. Yes. Yes, sir. Okay. All of you participants can go to the lab. Because you were doing real instruction yesterday, you were supposed to set up a PCI method. It wasn't much time to set up a PCI. Come back again here because Dr. Manoj Prasad will be on time. One thing I want to ask from the participants. Do you want to listen to a lecture? I'm not a deliver of lecture, but if you are interested, only I will tell you, otherwise I don't. Want to listen to me? Yes. But tomorrow morning, the schedule is very tight, so I can only deliver tomorrow morning from 8:30 to 9:30. Okay? Thanks. So to get up early tomorrow, make sure you're on time here.
Maybe for the lecture will be starting from 8:30. So maybe
Okay. Yeah,
you said yesterday.
oh, you don't have the agenda with you? Yeah. Okay. Okay. I am saying all the hours. I don't know. Because I write it, I'm not everything there. It's all about how long is the morning. Today morning, they say, you deliver only for 15 minutes. If you don't have time, you have to address it. Morning, you could say, little bit, maybe half an hour, that's it. Thank you, thank you. I don't
Question number. First,